Welcome back. For this section's activity, we'll be using a scientific method technique to study the concept of limiting factors. More specifically, how can space be a limiting factor for some organisms such as plants? We are going to need 3 disposable plastic cups, seeds, as well as soil. We will punch a hole in each cup to limit the amount of water retained by the soil. Then we will be labeling each cup with the amount of seeds they will have. As you may notice, the seeds we are using are just the uncooked, unfrozen beans we can get in the bag from the supermarket. Their seedlings will have thicker stems so they won't break and die that easily. Also, it is good to have the seeds out of the bag for a day or 2, kept in a dry place before planting them in the soil. They survive much better when they are dry. After planting the seeds, we're going to top off each cup to make sure the soil levels are the same. We're going to keep the cups in the same area, making sure they have the same environmental conditions in terms of sunlight and temperature. And we'll give each cup the same amount of water each day. The first seedling will appear by around day 6 or 7. This is when the real observations begin. You can use a camera to record your observations over the next 4 weeks. Following the scientific method steps, we can ask why plants don't grow that close to each other. And we can hypothesize that plants need space to grow properly. And we can predict that plants that grow too closely to each other won't be as healthy as those with more space. As we observe the growth of the plants and the amount of space needed by their roots and leaves, what conclusion can we make about space being a limiting factor for plants?